Creating a balanced portfolio. Well, there are various things which go into that, but of course I felt that I had to start off by telling you what the mechanism was for setting up a study. And of course, Mariel has set the scene beautifully with all those arrows. It looked like Agincourt, which is appropriate, arrows everywhere. I'll just go through the slide before I start on a diatribe. The first thing, of course, is you need to have a good idea, which in this situation means a good idea about a novel treatment, introduction of a previously ignored drug, whatever. And that idea has to be brought to the clinical studies group because we need to approve it before the trials unit. Does this thing work? Hopefully. Before the trials unit will start to work on it. And the trials unit are important mainly because they have the statistical knowledge that is needed for poor clinicians like me who can barely add 2 and 2 together to work out what size of groups we're going to need to evaluate whatever the good idea is. They will work with us to prepare the application for funding to the Clinical Trials Assessment and Advisory Committee, which is where we get our money from. Most of our money comes from CTAC. Once we have the money, then, of course, we have our ethics application. I do love it when people refer to the MHRA as a competent authority because, in my mind, that's something which they're not. Once we've got all that, then each individual trust where the trial study might be open will provide its own R&D approval, and some hospitals are extraordinarily slow. There is one that habitually has taken over a year from the moment it's been presented with the paperwork to actually having it open there. So then finally the studies get opened centre by centre, and it's a slow and painful process. And I've lost track. I think it must have been in 2008 that a grant application for the new acute lymphoblastic leukemia trial went in and was approved. And it was 2008, 2009, 2010. And although we call it 2011 now, it's actually only opened in the middle of 2012. And I've just entered patient number 3. Aren't I a good boy? But just to give you an idea about the complexity of the process which is now inflicted on us. What things do we need to balance? Well, unfortunately, we need to look at the cost. Sad but true. And in a public health perspective, pediatric cancer is a trivial problem. That is a statement of fact. And I'll talk a little bit more about that later on. But one of the unfortunate outcomes of our rearrangement has been that there's now no special consideration for pediatric cancer. We have to stand or fall on the merits of the study that we propose compared with everybody else's. And that leads to some significant difficulties. The likelihood of a successful outcome. How speculative can a study be? One of the consequences of the way all science is funded is that it tends to reward rather safe research rather than anything that's too speculative, because if it's very speculative, you might never get a useful answer. We need to have a balance of early, phase 1 versus late, phase 3 studies. We try and balance those studies which are looking at reduction of treatment against increasing it, so that's trying to balance the morbidity that excessive treatment costs against the mortality that's associated still with some conditions. And finally, there is always this argument about the frequency of a condition. So ALL is a very common disease, but with a very low mortality versus the number of children who might actually die from a particular condition, even though it's much rarer. And that, when you begin to look at things in that way, often causes a considerable reappraisal of importance. I wanted to talk about costs because I think it's important that people understand that the costs in running a trial are not related to the drugs that are used. As Mariel said, we have the EU Clinical Trials Directive, and that's is an EU directive which then is put into law in each individual country by whatever route is necessary. So in this country, it's the Clinical Trials Act, as amended 2006, I think it is now. And you can reflect on the fact that the Clinical Trials Act barely got a mention in Parliament, and we now have a great raft of legislation coming from the EU which barely figures in our democratic process. The next issue is, as I said, that paediatric cancer is rare. There's only 12 to 1,500 cases a year in the whole of the UK. And if you look at something like breast cancer, what is the incidence now? 1 in 7. So there's a huge disparity in the number of people affected. Moving along, as a minimum, a trial needs to be badged, and that means that the costs of treating a patient will be covered by the NHS for a phase 3 trial at any rate. What won't be covered, if that's the minimum achievement, are the actual administrative costs that are involved in gathering all the information in, analysing it, and so on. So ideally, it's fully funded for all the administrative costs as well. In general, early trials run by pharma, they will certainly give you the drug cost because they're likely to provide you with the drug for free and give you a substantial amount of money towards the administrative costs as well. But these tend to be very small studies, so that in the UK we're only recruiting maybe 2 or 3 or 4 patients to some of those studies. Speculation versus certainty. Well, when you devise, let's talk about a late phase study, so something which is going to be used for newly diagnosed patients, where, for the sake of argument, we want to improve the number of children who are cured. You need previous information from somewhere, and that might be from phase 1 and phase 2 studies, so that we know what the correct dose of the drug is, we know what kind of malignancies it's active against, and there may have been a small or even a fairly large single centre phase 3 study suggesting that this drug, in coordination with others, is particularly useful at improving survival. We need to satisfy the funding body, and as I've already said, the difficulty always is that funding bodies tend to like things which are safe. And it's little known but true that in what used to be called the MRC leukemia trials, because they'd often been done somewhere else previously, the experimental arm almost inevitably does better than the original arm. Now that perhaps indicates that we've done our homework, but also raises the question about how original are we being in our thinking. Funding bodies tend to be a stultifying influence. And finally, we do need to satisfy a research ethics committee, and I find this easier to live with, mostly, because I think it's appropriate that somebody who is completely disinterested should be able to say, the chances of that working, of that treatment being useful, are slender, and you shouldn't be doing it. I find that much easier to cope with than a funding body saying, that sounds a bit dodgy to me, I'm not sure we're going to get much publication out of that, we'll reject it. But that's just me. Clinical trial phases. I think this is important because there are lots of misconceptions, I find, about the way these early phase trials work and how new drugs might be useful. And I will speculate a little myself and say I doubt that a patient has ever been cured in an early phase trial. And the reason for that is that the drug company's view of a new drug is that it's a very dangerous compound which should be given to as few people as possible. Somebody who wants to have it might think this is the miracle compound which is going to cure me. But unfortunately, that's not the view of the drug company, and it's certainly not the view of the regulatory bodies that we have to answer to. So in phase 1, your dose finding. You start at a level that you know is safe. And that would mean that many patients might actually be getting a dose which is completely ineffective. The dose is gradually increased, usually something like 3 patients at a time at each dose level, until you know there are no toxicities. Eventually, you reach what's called the dose-limiting toxicity. So you might say you start at 10 milligrams per meter squared, work your way up 10, 30, 90, 150. You finally get to 200, and a patient has dose-limiting toxicity. You recruit another 3 patients, confirm the finding, and then you drop back 1 level. That's roughly the way a phase 1 study runs. And you'll appreciate, therefore, that the first 20 patients might have been getting a very low dose, which actually isn't going to be ever useful. In phase 2, you look at the range of activity, and you do that using the dose that you've derived in your phase 1 study. You test the drug across a range of likely tumours in the broadest sense. And again, to give you an idea about how the maths work out, at this stage, if 2 patients out of 42 had a positive response, that would be deemed encouraging. Well, the implication is quite clear that 40 of the 42 don't derive any benefit at all. Phase 3 is the sort of study that we're much more familiar with, where the drug is often used in combination, and your new combination of drugs then is used against the existing treatment. So some years ago, we tested dexamethasone against prednisolone in leukemia. The combination of drugs was otherwise identical. That was the only thing that we changed. And of course, there was a significant advantage to having the dexamethasone, the new drug, but then the trial had almost already been done anyway elsewhere. How did that get in twice? Have I gone the wrong way? Oh, am I going backwards? Hang on a minute. This is why I hate PCs. Hang on a minute, let me start from scratch. And then this is just to remind you that, obviously, early versus late. The early phase studies will only recruit a very few patients, maybe 1 or 2, because they'll often be run across the whole of Europe. Because we have a very slow system in this country, because the British like to gold plate EU directives when they enact the legislation, we often find that we're putting in the last couple of patients, and then the thing shuts. And that's, in fact, once or twice in the recent past, the study has shut before the Brits have managed to recruit anybody. It is often pharma-sponsored. The late phase study will recruit many patients. Leukemia studies routinely recruit over 2,000 patients just in the UK. It will usually be for initial therapy, first-line therapy. It will run over a period of years, 5 years, 7 years, maybe even 10 years, and most likely have an academic sponsor. Let's see if it behaves this time. I don't trust this. That's right. That's right, isn't it? Yes, it is going forward. I talked about the frequency of the condition versus the mortality, and historically, people have focused on things which are very, very common. So ALL has always had a study, and in fact, the Medical Research Council first set up a childhood leukemia working party, which was chaired by the father of my now retired colleague in Oxford, Sir Alan Moncrief, chaired the original MRC working party. And that was, I think, in 1956. People barely knew what leukemia was. Other studies have been more recent in their initiation, and I listened recently to Alan Kraft talking about the genesis of what was then the UK CCSG, and there were 8 of them, I think he said, sitting in Birmingham having a discussion. And at the end, everybody had forgotten about Alan, and the only thing that had been left behind was bone tumour, so he got bones. Which is a curious way of allocating roles. Here we are, ALL is common, but has a very low mortality rate. AML is much rarer and has a higher mortality rate. And across the country, if you look at how important, if you rate something in terms of how many children actually die, then you might be much more interested in acute myeloid leukemia than in ALL, that some people already would say is a boring condition. We should hoist the flag of victory and all go home. Some sympathy with that. Many brain tumours are rare, but they have not only a high mortality, but also a very marked morbidity. And I have every sympathy with the central nervous system tumour group, who constantly remind us of this fact, ceaselessly actually. But I do have a lot of sympathy with their point of view. Possibly the most difficult thing to deal with is the morbidity of so-called successful brain tumour treatment. It is now very difficult to establish trials for rare conditions because of the nature of the statistical process, the lack of prior information, the lack of interest of drug companies, because they're never going to make their money back, and they are, after all, money-making organizations, really. And so it's very, very difficult to establish trials for rare conditions. The other problem being, nobody's really got a good idea. And if you don't have a good idea, you're stuck at the very beginning. And that's often a problem, I'm afraid. So in summary, I've given you a quick run through the sort of things we try to take into account. What I hope we've achieved over the last, or are achieving, hopefully by the end of this year, is that we will have an open trial for all the major tumour groups for initial therapy that will bear the scrutiny of time, produce useful results, is properly funded, statistically well-founded, and so on. We are increasingly developing studies for patients with relapse, and we now have, I think, actually quite a steady throughput of early phase trials to evaluate new drugs. I hope that comes across as a balanced portfolio, taking into account all the things I've alluded to. Thank you.